Hello everybody, and thank you for joining us today. I'm Simon Lightfoot from BioFPR, and I'm here to introduce this web seminar entitled Feedstock Characterization and Fermentation Monitoring of Biofuels. Today's webinar is brought to you by Thermo Scientific. We're delighted to welcome three excellent speakers today. Paul Volcker is Manager of Global Market Development for the Chemicals and Petrochemicals market at Thermo Fisher Scientific. He works with key academic and industry experts to develop improvements in chemical and petrochemical analysis methods. He has over 10 years market and applications experience in the areas of proteomic, genomic, and cellular separation and analysis processes. Paul holds an MS and BS in chemistry from UC Davis and California State University at Hayward, respectively. Archana Pandey is a Senior Research Associate in the Analytical R&D Lab at LS9, a biotech firm based in South San Francisco. She works on analytical method development and validation for detection and quantification of natural product compounds, performing assay and analysis of target compounds in the analytical lab, and provides analytical support to different scientific staff and projects utilizing various analytical techniques to extract and accurately quantify small molecules in high cell density fermentation. Archana has a master's in analytical chemistry from Rochester Institute of Technology. Before joining LS9, she worked in the area of good laboratory and manufacturing processes in the pharmaceutical industry. Kevin Chambliss is an associate professor of chemistry at Baylor University, Texas. He is recognized as an authority in the field of chemical separations and analysis, in particular, solvent extraction and ion exchange technologies that remove pollutants in natural waters. Before joining the Baylor faculty in 2001, he served as a postdoctoral fellow at Oak Ridge National Laboratory and as an assistant professor at Mercer University. During this time, he investigated strategies for cleaning up nuclear process waste at several Department of Energy sites. Kevin has presented more than 20 papers on his research findings. His current research focuses on the design, synthesis, and characterization of novel compounds where molecules recognize environmental contaminants and medical targets. A very warm welcome to you all. Now, before we get started, just let me remind you that you can ask questions anytime during the presentation by typing your question into the Q&A panel on the page that you should be viewing now. Your question will not be seen by other attendees, so don't be shy about asking them. Questions will be answered during the Q&A session after all three presentations have been given. Any questions that we don't get to today, the panel will promise to do their best to follow up with you via email over the coming days. And so at this time, I would like to turn the floor over to our first speaker, Paul Volcker. Paul, it's over to you.
Thank you, Simon. This first talk provides an introduction to today's presentations with an overview of the operating principles of HPAE-PAD, or high-pressure anion exchange chromatography with pulse amperometric detection. It also touches on why certain detectors, columns, and ion chromatography, or IC systems, were selected over others, and how they work together as a complete system to give you the accuracy and reproducibility you expect. For those of you who are new to amperometry, let me mention a few key points to get you up to speed on this electrochemical detector. Amperometry measures current or charge resulting from the oxidation or reduction of an analyte at the surface of an electrode. Oxidation is when an electron goes from an analyte to the electrode, and reduction is when the electron goes from the electrode to the analyte. The current produced is measured by the detector. When a single potential is applied to the electrode, the detection method is DC amperometry. Pulse amperometry uses a repeating sequence of potentials, or waveforms, to enable detection of analytes on metal electrodes such as gold, platinum, and silver, making it more sensitive than DC amperometry. Another important advantage of PAD is its high selectivity. Using PAD and gold electrodes, non-derivatized carbohydrates and amino acids, for example, can be analyzed in the presence of high concentrations of other analytes. In addition, PAD can detect carbohydrates of up to 10,000 Daltons. This slide shows the oxidation reaction for glucose with PAD. As the sugar is oxidized, it generates electrons. It is this charge that is picked up by the detector and is proportional to the concentration of glucose, or oligosaccharide that is being oxidized at the electrode. Two key points can be made from this statement. The first is the reaction generates hydrogen ions and is therefore driven by high pH. This makes PAD an ideal companion for HPAE as the separations are done at high pH. The second is PAD does not oxidize all the carbohydrate present, only about 1%. It is therefore compatible with preparative chromatography and it's compatible with mass spectrometry since 99% of the material will be left intact. Shown here is the family of CarboPack columns. There are several types of columns depending on your application needs. For this webinar, we will focus on the columns appearing on the upper left-hand corner. That is the CarboPack PA10, PA20, and S8 designed for separating mono and disaccharides. A PA10 and PA20 belong to the same class of column type, a non-porous bead coated with fine latex. PA10 is optimized to determine amino, neutral, and acidic monosaccharides. The PA20 consists of smaller particle size to provide higher resolution and with the shorter column format enables faster separations. The S8 is composed of a macroporous bead coated with fine latex. It has significantly higher capacity than other CarboPack columns and is the column of choice for high concentration mono and disaccharides. As an example, a 150 gram per liter corn stover hydrolysate sample was separated with high resolution after only a 1 to 200 dilution. And it is fast. In less than 10 minutes, it separates the 8 most common biofuel mono and disaccharides. The final component of the HPAE-PAD is the IC system. Shown here is the ICS 5000. Now, why the ICS 5000? It is a metal-free IC system, a requirement for using PAD, and the only compatible model that can be quoted with PAD. In conclusion, the ICS 5000, CarboPack columns, and PAD form a complete system that provides direct quantitation of non-derivatized carbohydrates with minimal sample preparation or cleanup. Thank you. So without further delay, let me turn the floor over to our next presenter, Archana Pandey of LS9 Incorporated. Archana?
Thanks, Paul. Hello, everybody. First off, I would like to thank Paul for giving me this opportunity to present, to participate in this webinar, and thanks to everybody for joining the webinar. In this session, I'm going to talk about feedstock analysis of mono and disaccharides using HPAE chromatography coupled with pulse electrochemical detection with an online event generator. This is very much in relation to the application in rapidly growing biofuel or even chemical industry where we don't have a fast and robust approach for analysis of all sugars in complex biomass. Now, there are several LC methods in the industry for analysis of different kinds of sugars. However, development of a robust analytical approach for accurate quantification of all mono and disaccharides and oligomers in cellulosic biofuel feedstocks can be really very challenging. Typically, method utilizing ion exchange chromatography with silica-based or polymer-based cation exchange column using an RI or UV detection for sugars are not selective for all mono and disaccharides in feedstock, and they are further limited by matrix interferences arising from complex biological matrix. High-performance anion exchange chromatography coupled to PAD using an online event generator is a very effective technology today to determine all the sugars in concentrated feedstock and high cell density fermentation samples encountered in the biofuel industry. So when we first got the Dionex ICS 5000 system installed in our facility, sometimes this April, the objective was very simple. The objective was to separate all these sugars, all these 8 sugars, given our feedstock has these sugars at different concentration, and then establish dynamic range of the method once we have the optimal separation. So I first started working on CarboPack PA20 column and then eventually switched to S8 column based on our application needs. Now, once we know the dynamic range of the method, we wanted to determine the accuracy and reproducibility over this range, and finally the goal was to be able to analyze concentrated feedstock sample and start monitoring all sugars in fermentation samples. Again, not to mention all this with high confidence in the accuracy of the numbers being reported because our yield calculation was based on these numbers. Prior to working on ICS 5000 dynamic system, we were analyzing sugars on traditional LC system with refractive index detector. Traditional LC method with carbomix calcium-based column can separate only 4 sugars, glucose, fructose, sucrose, and xylose, as you can see in this slide, in 20 minutes. Overall method runtime, however, was 25 to 30 minutes to avoid any carryovers in the subsequent run while running real samples. So the point here is that not only the method runtime is too long here, with only separating 4 sugars, the method also has limitation to separation of other components in the feedstock. Arabinose, as you can see here, coelutes with fructose, and mannose and galactose coelutes with xylose. So we transition from traditional LC to Tandem LC column, which is basically 2 carbomix lead-based LC column in tandem. Using Tandem column, we can separate 3 additional sugars, cellobiose, galactose, and mannose, other than glucose and xylose. So basically, we can separate total of 5 sugars here, cellobiose, glucose, galactose, xylose, and mannose on this Tandem column with RA detector, with refractive index detector. Not to mention, these 5 sugars take 45 minutes to resolve on the Tandem column, and we still have issues separating arabinose and fructose from the rest of the sugars. This is when we transition from Tandem LC to dynamic system. I started to work on PA20 column, and it was really very encouraging to see the separation of 6 monosaccharides on this column in less than 20 minutes, as you can see here in the top slide. So the question is, what is different? Why and how the IC is different from traditional LC system? Now, unlike traditional LC system, ion chromatography HPAE-PAD takes advantage of weakly acidic nature of carbohydrate, where the detection is based on measuring current resulting from the oxidation of carbohydrate at the surface of the gold electrode. At high pH, application of positive potential oxidizes the carbohydrate at the surface of the electrode and separates using anion exchange stationary phase coupled with eluent generator, which produces carbonate-free hydroxide eluent to ensure reproducible and enhanced separation. Whereas in traditional silica-based columns, the matrices dissolve at high pH, limiting the resolution of all sugars. So it was really very exciting to see the separation of most of the sugars on this PA20 column. Now, given our sample profile and column specs, wash step was recommended, and when I introduced the wash step, I observed cellobiose eluting towards the end of the run, as you can see in the bottom slide. So the method runtime was elongated from 20 to 45 minutes, which was not very encouraging at that time, and all the more, oligomers were still a problem to separate even with such elongated method runtime. At that time, and even otherwise, what mattered most to us was resolution and accurate quantification, very accurate quantification of the desired sugar in the feedstock sample. And since I was able to get an optimal separation of 6 monosaccharides on this PA20 column in less than 20 minutes, I started working on this column to understand the advantages and limitations of this column and the method. I calibrated the method. We had a quadratic fit with R-square of 3 nines over dynamic range of 2.5 ppm to 80 ppm. So the linearity looked good. Sensitivity was great at lower end. However, I still could not go above 80 ppm, which was not ideal for our application. Also, ideally, you would like to go for a linear fit. And while we were aiming for that, we could not achieve a linear fit on this column, specifically with the range we were looking at. Since we were also interested in oligomers, while seeing the possibility of reducing the method runtime and obtaining linear fit, all this lead for further method optimization. Every time we run our sample, we monitor our controls using check standard, which tell us how fit our curve is based on how stable our system is performing that particular day and how accurate our numbers are on real samples. So following calibration, I prepared and made 6 injection of QC, which was glucose at a concentration of 50 ppm, to check the accuracy and precision of the method on this PA20 column. The accuracy between different runs on different days was between 93 to 103%, which is plus minus 5 to 7%, and RSD was less than 3%. Again, this QC was run on PA20 column and quantified using quadratic fit. Obviously, we were looking for a better accuracy. Here is a sample, here's a profile of the real sample, which was diluted 10,000-fold to be in the dynamic range of the method. Now, such high dilution was not realistic, was not feasible with the number of samples we were expecting to analyze, and also with such high dilution, it was highly likely that we are more prone to introducing significant error in our calculation. Nevertheless, we could not separate oligomers, and as I keep repeating, as you can see in the chromatogram, hence the need for method optimization still continued. Now here's a comparison of feedstock analysis with lot X, which is lot of cellulosic feedstock obtained from external source. The percent composition of individual sugars in this feedstock sample is compared with the established composition given for lot X on this PA20 column, all based on weight per weight. Now some numbers were close, as you can see, and some are not. Again, since the accuracy on QC was in question, was because of the quadratic fit was not that great, we were not sure if the error is because of the error in dilution or we need to further optimize the method. Now our application is very different from pharma. Our feedstock sample is a very concentrated sample, and to analyze the same, we cannot ignore dilution. Now given the different range of sugar concentration we have in our feedstock, and given the dynamic range of the method and also the sensitivity on the instrument, we need to plan the dilution in such a way such that the accuracy is not compromised at any level. Likewise, the fermentation broth sample also has to be diluted to do accurate quantification of each component in the broth sample. Now, the most accurate way of doing dilution, as you all will agree with me, is using volumetric flask and using volumetric pipette dilution. However, it is highly impractical to do volumetric dilution on all the samples. Hence, it was very important for us to understand the variability in numbers from doing dilution using electronic versus volumetric pipette. So what I did here is I prepared a concentrated solution of feedstock around 10 to 12 grams per liter, did 100-fold dilution of this feedstock using volumetric pipette on the top slide to get 100 mg per liter, and made 3 injection of this volumetric dilution on PA20 column. Similarly, I also did 100-fold dilution of this concentrated feedstock using electronic pipette to get same concentration of 100 mg per liter, and this dilution was done in triplicate, and each rep was injected in duplicate to have a total of 6 injections on this column from electronic dilution. Now, average of the numbers from electronic and volumetric dilution was then actually compared. Now, here's the result from the analysis. The analysis was done for lot X over a period of 3 consecutive days, and what is reported here is the average of the reps. Again, at this time, I'm not comparing the result between my numbers and lot X. All I'm looking for is the variability between dilution from volumetric and electronic pipette. As you can see here, that internally the numbers are very close, with less than 1 to 2% variability between dilutions using electronic and volumetric pipette. Now, there are some discrepancies in actual numbers for certain compounds when we compare with lot X, and while we were trying to figure this out, it was very clear that we can do electronic dilution going forward for all the samples. Now here is the analysis of unknown mix of sugar from external source. What is done here is basically a blind test, which was another way of confirming if our method was robust and in sync with the external lab. The 2 samples given from external lab, sample 1 and sample 2, they were at different concentration, and different dilution was done by me for these samples using electronic pipette to be in the dynamic range of the method and injected on PA20 column. For both the samples, sample 1 and sample 2, as you can see in the bottom slide, the numbers for each sugar very well matched with the external lab. Now, what I have been talking so far is the work done on PA20 column. We certainly had some limitations on this column in terms of method runtime, dynamic range, and we also believed that accuracy could be better. So the major driver for transitioning to S8 column was to reduce the method runtime, increase the range so we could further reduce the dilutions, and most importantly, increase the accuracy by obtaining a linear fit. So basically, we were hoping to have everything, have a better accuracy with less dilution over desired dynamic range. 2 things that really made a significant difference in our application and analysis was introducing 0.4 microliter internal lobe and a thicker gasket in addition to the S8 column. Earlier, I was using an external 25 microliter loop installed on the valve. This is basically a coil of back pressure tubing installed between EG and injection valve, which is very common during installation. The 0.4 microliter internal loop is a special valve spot in its own, as it would be practically impossible to get a tubing narrow enough to make it 0.4 microliter are long enough to connect between the ports. So this was supposed to help load more on the column by decreasing the sensitivity. Also, I transitioned from 2 mil gasket to 15 mil gasket, which pretty much did the same thing. Increasing gasket thickness also helped decrease detector sensitivity and allowed to inject more concentrated sample without the need for larger dilution. In the nutshell, introducing S8 column and reducing the injection volume to 0.4 microliter with internal injection valve pod and decreasing the detector sensitivity with thicker gasket helped increase the dynamic range of the method, provided linear fit, and allowed to load more on the column with less dilution. In addition to all this, introducing internal standard sucrose was also a very important step taken at our end to control injection variability. Here is a separation of all mono and disaccharide on CarboPack S8 column, which was achieved in less than 10 minutes with no additional wash step required. So it was really amazing to see all the compounds I have been trying to separate forever and quantify in my feedstock sample was so very well separated and resolved in less than 10 minutes. And even better, we were also able to achieve a linear fit with R-square of 3 nines on this S8 column for all sugars. So basically, I got everything I was looking for. But as I mentioned before, our feedstock sample is a very concentrated sample which has both mono and disaccharide in different range of sugar concentration. Certain sugars are as high as 40 to 50% of total sugar concentration, and some sugar are as low as 1 to 2% of total sugar concentration, all based on weight per weight. So hence we needed to define a range where we can have a good accuracy at both the levels, at all the levels. However, at the same time, we needed to strike a balance between practicality and accuracy. So in all likelihood, in all possible scenarios, the best range that we could possibly establish on S8 column with good linear fit, great accuracy over desired dynamic range to meet our application need was 5 ppm to 300 ppm. We probably could have gone even higher, as high as 500 ppm or even more to achieve the upper limit on the column so that we need to do even lesser dilution. However, in such case, the linearity was established with an R-square of 2 nines only, which impacted our accuracy to some extent, and our apprehension was that our accuracy at the lower end will be significantly compromised. Now, given that we have certain sugars in the feedstock at a very low concentration, we were not comfortable with that, and we decided to stick to this dynamic range. Here's the accuracy on standards when standards are processed as samples. The accuracy on standards for all the levels was actually greater than 98%. Same QC, that is glucose at 50 ppm, which on PA20 column with quadratic fit gave an accuracy plus or minus 5 to 7%, when run on S8 column with linear fit gave much more tighter accuracy, 98 to 102%, that is within variability of plus or minus 1 to 2%. The reproducibility, the RST, was also very tight, less than 1.5%. This QC was prepared to accommodate the range, keeping in mind the established range on the older method using PA20 column. However, once internal standard fucose was introduced in the new method on S8 column with a newer range, another QC was prepared with glucose and mannose at concentration of 100 ppm, each with internal standard, and the accuracy on this new QC has always been greater than 98%. In this slide, you can see it's almost like 100%. Here is a profile of feedstock and fermentation broth sample. On this S8 column with modified method, 1,000-fold dilution of feedstock was done instead of 10,000 or 100,000-fold dilution I was doing in PA20 column, and 20 to 50-fold dilution of fermentation broth sample was done to achieve the separation and to achieve the desired results with good accuracy on numbers. Feedstock analysis from same lot X was done again and was analyzed now on S8 column. This was done repeatedly for 3 consecutive days, and the numbers not only very well agreed to the certificate of analysis established for lot X, but most importantly, as you can see here, the numbers are also very consistent in inter-day analysis. Now, fructose values internally, they were very good, they were very tight. However, they are lower than the value reported for lot XC of A, and we are still in the process of figuring this out. Now, with this, I would like to wrap up my presentation here by concluding in this slide So after working on Dynamics ICS 5000 system for more than 6 months now, I can confidently say that the objective of optimizing the method by establishing good accuracy and reproducibility over desired dynamic range while meeting our application need has been successfully accomplished, and today I'm doing routine analysis of all feedstock and fermentation samples and reporting results with high confidence in the number. With this, I would like to pass on the floor to Kevin, the next presenter. Kevin?
Thanks very much. So I think the topic that I'm going to talk about today is also quite timely. Following on our last presentation, it's related to the same subject where we're talking about rapid and selective determination of carbohydrates in samples that are relevant to biofuels R&D using high-performance anion exchange with pulsed amperometric detection. And building on our last presentation, the content of my talk today will be a little bit about the historical development of the SA10 stationary phase, and I'll also provide an interlab comparison of reproducibility for a variety of different sample types to give you an idea of the level of performance that could be expected if you decide to use the method. So first, let's talk just briefly about the types of analyses that are typically performed in biofuels R&D. While the final stage of any sort of production paradigm is still a subject of debate and still undergoes considerable research, most processes under investigation take a feedstock through a three-step procedure, the first being chemical pretreatment, which is designed to expose cellulose within the material to enzymes, which can then hydrolyze cellulose down to monomeric sugars, and ultimately those can be fermented to product, be it a biofuel or some other type of chemical. Along this process change, there's several places where monitoring sugars is an important thing to do. First and foremost, people are interested in total sugar content of a feedstock because this ultimately gives them the starting number that lets them scale to a potential product and how much of that they could produce at any given time. In this case, you're typically measuring total sugar. That's done by a two-stage sulfuric acid hydrolysis of the feedstock. They're measured as monomers. And this procedure is called quantitative sakurafication. There are standard protocols for performing that available at NRO. Also in feedstocks, it's become increasingly interesting for some labs to begin to look at what's the content of aqueous extracts in the feedstock. So it's just a simple aqueous extraction. In this case, researchers are interested not only in total sugar, but also in free sugar, which includes monomers plus sucrose. Most aqueous extracts can contain primarily sucrose, glucose, and fructose. Following pretreatment or the initial hydrolysis chemistry that goes onto the feedstock, people can also be interested in free and total sugar. Similar analyses are of interest in the enzymatic hydrolysis step, and then free sugar is typically monitored in fermentation broths, as we just learned in our last presentation. So as we keep that in mind, what you need to recognize is there are a variety of different places we'd like to look at carbohydrates in the biomass to bioproduct production paradigm. The samples of interest are highly complex, and what I hope to convince you of is that the HPAE-PAD approach is got some real advantages relative to RI detection beyond simply the improved chromatography that can be expected. So while a number of methods exist, current practice relies heavily on the RI approach. In combination with ligand exchange chromatography, we saw a similar chromatogram in the last presentation, and here I'm showing separation of the target analytes that are typically of interest in biofuels R&D being separated on a lead form Shodex column. The eluent in this case is pure water at 0.6 mils per minute. And although the sugars are separated between 12 and 22 minutes, run times with this approach actually approach 60 minutes. Typically, there's a 30 to 35 minute hold beyond dilution of the last sugar that's required to elute all the other components of the sample. As we saw in the last presentation, There are typically some problems with co-eluting analytes. In this particular case, you can see that there's some overlap in arabinose and mannose. But even more significantly is that sucrose and cellobiose co-elute under this paradigm. If we think back to the types of samples that need to be analyzed from sort of the introduction I gave in the last slide, it's important to recognize that mannose is typically not present in samples unless we're talking about aqueous extracts. And sucrose, that's also true. So it may not be as critical to separate these for a given application. Nevertheless, it's always better if we can be certain that there's no problems due to coelution. The reason the RI approach has been so widely utilized is that it's proven to be robust. It's proven to be very easy for people to use. The wide dynamic range of RI is compatible with the wide variability that you can get in sugars in these types of analyses. And it also offers the possibility to look at some other important compounds, for example, things like acetic acid, furfural, and HMF, which are important inhibitors in these processes. So there's pros and there's some cons. The biggest cons, just to summarize, are poor elution of select analytes and long run times. So a little over a year ago, our laboratory reported in Journal Chromatography a significant improvement in runtime and resolution using a carbonate modified CarboPack PA20 column. And the chromatogram shown here is for a CarboPack PA20 after modification. Modification was typically accomplished by sequential injections of sodium carbonate. And what we observed is that the modification procedure primarily affected the retention time of sucrose. If you looked at this same separation on a PA20 that was not modified, sucrose would co-elute with galactose, and cellobiose and maltose would obviously elute at much longer retention times, much like we saw in the last presentation. So modification with carbonate enabled us to resolve sucrose. It turns out that temperature is also a really important component of this separation, and so the blue square shows the peaks that are most sensitive to temperature. In most applications, if you're looking at aqueous extracts, there's not a lot of xylose. And in that case, if you go to higher temperature, you can move the mannose peak closer to where xylose is shown here and get it resolved from fructose. If you're looking at hydrolysates, typically you don't see a lot of mannose, and so there just xylose and fructose become important. But we thought this was a pretty significant advance, although the paper, which is referenced here, I believe shows some pretty convincing data that repeated modifications result in comparable column performance. Obviously, in an industrial type setting, the need to modify a column is not ideal. As we saw in the last presentation, the other caveat of this approach is primarily due to the inherent sensitivity of pulsed amperometric detection. Here I'm showing some quantitative Sakurafication samples from corn stover, switchgrass, and poplar, and you can see that the dilution factors range from 400 to 1,000. Other data reported in that paper required a 1 to 2,000 dilution factor in order to bring things within the linear range of the pulsed amperometric detector. And for reasons discussed in the last presentation, we all know that this is not optimal. So inspired by the approach that we observed with the carbonate modified PA20, Chris Pohl and others at Dionex went back to the R&D table and developed the SA10 stationary phase, which offered comparable runtime and resolution to what was observed with the modified PA20 column. Alleviating that need for modification is an obvious benefit, and here, if you're careful at looking at slides, you notice that the other one has an equivalent concentration, but there's a higher flow rate involved. This is simply reflective of the increased capacity of the SA10 stationary phase compared to the PA20. In addition to alleviating the need for modification, scientists at Dionex or at Thermo now Also engineered two hardware developments that were mentioned in the last case that have addressed the dilution requirements affiliated with pulse tamperometric detection. The first that we talked about a little bit is going from a 10 microliter in the previous presentation, 25 microliter sample loop to a 400 nanoliter internal sample loop. This is a Reodyne valve, and it's shown in the lower right portion of the slide here. Also, in the particular application that I'm going to talk about today, we replaced a standard 2 mil spacer with a 62 mil gasket. And so you can see the picture in the upper right compares the width of those two gaskets that ultimately define the volume of the electrochemical cell where pulse tamperometry is conducted. And these two factors work in concert to reduce the sensitivity as we saw previously, and I'll show you some more data about that in a couple of slides. So with that introduction, what I'd like to do today is talk to you about some work that we've been doing over the last few months in collaboration with Thermo and with scientists at the National Renewable Energy Lab. I'm happy today to get to be the messenger in this presentation, but I certainly want to acknowledge the people that have done the heavy lifting and the hard work in data collection and in operation of the instrumentation, and they're shown on the slide here. In this particular study, all instrument configurations were ICS 3000 systems, which don't want to have any source of confusion for anyone. This was simply the precursor to the ICS 5000. It's also a peak system and is quite comparable. Each instrument in all three laboratories was equipped with an eluent generator, an auto sampler, the low volume injection valve that I talked about on the last slide, a SA10 column, and the large volume spacer in the detection cell. And while there's a lot of things that we could do with the data I'm going to show you today, I have three primary objectives. The first is to talk a little more about the linear range of the PAD detector. I then want to show you a reproducibility comparison between labs with real samples, and lastly talk a little bit about concentrations determined using the PAD approach compared to what you would see with the method that more commonly is utilized for these types of determinations. So first, let's talk a little bit about linear range. I think most of us in practice are used to gauging linearity by looking at an R-square value for a calibration curve. In fact, if we really want to get down to the nuts and bolts of a linear response, it's a little more accurate to look at a response factor and begin to see what deviations you get from a stable response factor as a function of concentration. And so that's the plot I'm showing here at the beginning. And you see that the response factor basically decreases as you go from very low to higher concentrations. In trying to define linearity, what we did here is find a good mean where it was roughly linear, and we said we were willing to accept a 5% deviation on either side of that mean. And in doing that, what you basically observe is that the linear range of the detector spans at least 1 to 1.5 orders of magnitude. And before anyone gets upset at that and says, well, no, it's actually much higher, I will obviously say that you could extend this range and probably still get a decent R-square value. But we actually viewed this as good news. If you look into the details of pulse tamperometry, a limited dynamic range compared to some other detectors is common for that. About 1.5 orders of magnitude is what most people would expect. And what this suggested to us is that using the wider spacer in the flow cell didn't compromise the basic operating principles we would expect out of PAD. If you choose that linear range and you make a typical calibration curve, you can see in the bottom view graph that between about 0.02 grams per liter and up to 3 grams per liter, you can get very linear calibration responses. And ultimately, that's about the range that we looked at in our interlab comparison. In fact, I believe we didn't go all the way to 3 for the rest of the data that I'm going to show you. I believe the low end of that range is correct, but the upper end should be about 2.4 grams per liter. So once we did that, we wanted to compare calibration sensitivity at each of the 3 sites. And By calibration sensitivity, what I'm really talking about is the slope of the calibration plot. And what you can see is for one of the sugars we expect to be present in highest concentration, glucose, as well as one of the more minor sugars or sugar present at lower concentration, galactose, you see that we get very good agreement in calibration sensitivity between the three laboratories. If you continue to look down, I'm just showing data from site 1 for xylose through cellobiose. You see that there's some variability in the slope that you get, and that the sensitivity of every sugar is not identical, but they're all within about a factor of 2. The intercepts in this case are close to 0. I will point out that if you have an astute eye, there appears to be some positive bias in this data, and that in fact the negative value shown as an intercept for cellobiose was the only negative value we saw in the study. And we basically interpret that to mean that since we're pushing the upper end of that linear range, if we are getting out to the point where it begins to bend away from linearity, that would bias the slope positive a little bit. And so we believe that's probably the most logical explanation for that. In all cases, we got at least 2 nines in R-square values for what was observed. And then I want to talk for just a minute about the quantitation limits that can be observed in this approach. And so I'm showing LOQ values in column 4 of this particular table, and this would be LOQ determined at a signal-to-noise ratio of 10. In this case, you see that we're reporting them in units of 10 to the second grams per liter. For comparative purposes, if we think back to the detection limits or quantitation limits that we reported in our subject et al. Study, where we used a standard 10 microliter injection and a 2 mil spacer, to get sort of the same whole number type report there, you would need to think about 10 to the first micrograms per liter. And so the combined effect of the low volume injector and the larger spacer resulted in about a 4 to 5 order of magnitude decrease in sensitivity for the PAD detector. And so we've come a long way in alleviating that dilution requirement. And here's just kind of a quantitative comparison of that. So for our reproducibility study, we wanted to look at real samples. We took 21 opportunistic R&D samples that were available in NREL. These samples represented quantitative Sakurafication samples or samples from feedstock compositional analysis with 2 different potential feedstocks, corn stover and miscanthus, as well as a standard reference material available from NIST. We looked at 8 different pretreatment samples. Basically, we looked at 4 pretreatments, 2 with the vertical reactor, 1 with the steam gun, 1 with the slurry reactor. And the 8 samples comprised 4 that would be used to determine free or monomeric sugar, and then 4 from those same pretreatments for total sugar. We looked at 8 different samples from simultaneous Sakurafication and fermentation, 3 at T equals 0, 5 at the conclusion of that experiment, and then had 1 synthetic sample as a control. And as you can imagine, you might want to go in and think about the data and how different samples affected different variables within the methodology. I'm not going to go to that level of detail today. I just want you to recognize that the samples that we analyzed to generate the data I'm going to show you were representative of what might be expected in R&D activities. At the far right, you can see the sample dilution that was required to bring most analytes within the linear range. In fact, xylose exceeded the linear range that was calibrated in a few samples, and all data that I show you excludes those. So we're only talking about data that was quantified within the calibrated range of the detector for the study. So I think most people will be familiar with what's called a QQ plot. In each of the plots that I'm going to show you on the next few slides, what you're seeing is concentration of sugar on the X and Y axis. In this particular case, if we look at the upper left, we're seeing concentration of glucose determined in those 21 samples at site 1. It's compared with the concentration at site 2. In a perfect world, the slope of this correlation would be 1, and in fact it was for that particular case. If you make the same comparison in glucose concentrations between site 1 and site 3, we see some deviation from the expected value of 1 in a positive direction. In contrast, if we look now to the right-hand side of this slide, you're seeing the same sort of plots for xylose. And again, we don't get a perfect correlation, but in general, the data look pretty good in terms of reproducibility that you could expect for these two sugars that are typically present at highest concentrations in samples. We also did the same type of comparison for sugars present at lower concentration. The data for arabinose look quite good. For galactose, at least for the Site 1 and Site 3 comparison, they're not quite as compelling. However, if you look beyond the slope, you see that here we're measuring sugars between 2 and 3 grams per liter in concentration, and at both sites you observe that. The correlation analysis here in QQ is probably not nearly as compelling for galactose or even arabinose as it was for glucose and xylose, because the concentrations of these sugars don't span nearly as large of a range. And nevertheless, the data are as they are. We also observed fructose in about 4 samples, but the concentrations were sufficiently similar that it's not really even a good comparison for me to show you that. So we then want to move on and talk a little bit about how does the HPAE method compare with HPLC-RI. And here again, I'm showing you QQ plots for concentrations determined in these same samples via the PAD approach, as well as those with RI on the y-axis. And again, you see that the correlation between the two methods is similar to what we observed for interlab reproducibility. If we want to look closely, it's obvious that as sugar concentrations get higher, there appears to be a little bit of high bias in the PAD data relative to the RI data. Is that important or not? Probably depends on who we're talking to, but nevertheless, for the sake of argument, I think it's worth thinking about for just a moment. If we looked at these data for minor sugars, which I don't have a view graph for, the deviations were much larger, and they were in both positive and negative directions. I would offer at this point that co-eluting species with refractive index detection can cause erroneous information in both positive and negative directions. And for those that are big fans of the RI approach, you may be wondering why I'm looking at a QQ plot and sort of blaming RI as the potential bad actor here. Paul's already talked to us about the inherited selectivity advantages of PAD relative to RI, and obviously I will say that we're not going to learn a lot just from looking at QQ plots. So I want to show one busy slide that gives you some raw data from this study, and what I've done is select 3 samples, and I'm showing you data from sites 1, 2, and 3 that were determined with the PAD approach. I'm also showing you the raw data from the RI approach. Before we talk about that specifically, I'll also note that the values in parentheses represent one standard deviation in the least significant figure. You can see that the precision that may be expected between the two methods is pretty much on par. Typically, we have uncertainty in the 2nd or 3rd decimal point. So back to the point at hand, what I want you to see in red is that for the sample shown at the top of this slide, so the first sample, we actually get detections for arabinose, galactose, fructose, and cellobiose with the RI approach. However, none of the 3 labs using the PAD approach observed a peak at retention times corresponding to those analytes. If we then move down and look at the second sample, the important thing for you to notice here is that we did have some samples where those sugars were detected, and where they're detected, although the concentrations between the PAD approach and the RI approach are comparable, the most important thing I want you to see is that in the sample shown in the middle here, the concentrations that were observed with the PAD approach are well below the concentrations in sample 1, where we did not observe anything with the PAD approach. And just so people know that I didn't grab 2 samples to show something, I'm also showing a 3rd sample that shows the same trend. And so each number in red represents a false positive detection of a carbohydrate when the RI approach is used. The frequency numbers at the bottom of the slide represent the number of samples out of 21 analyzed where a given sugar showed up as a false positive with this type of analysis. And so As many as 12 of 21 samples had false positives for galactose. Xylose, we didn't observe any, but we did see even 4 false positives for glucose in this particular analysis. So that's about all the time that I'm going to have to show you what we've been doing today. I will make some concluding remarks, and I hope that collectively the presentations that you've heard today have demonstrated that the anion exchange chromatography PAD approach is superior to HPLC-RI for determination of sugars in biomass samples. To recap, the SA10 stationary phase offers significant improvements in runtime and resolution, especially of sucrose, which wasn't that important in the study that I talked about today, but for anyone interested in aqueous extracts, sucrose is very important. The interlab comparison with a novel method that uses the SA10 in combination with a low-volume injector and high-volume detection cell proved to be robust and reproducible in our interlab comparison. The hardware modifications that were engineered at Thermo Fisher certainly have reduced the dilution requirement to a point where it becomes reasonable. And perhaps the most important thing in my presentation today, which I didn't necessarily mean to leave the cursor here, but I think it's a great take-home point to leave us with, is that if you routinely use the RI approach, false positives are likely. And this is most important when you're interested in concentrations of some of the sugars present at low concentration. So with that, I'll thank everybody for their time. I appreciate you attending, and I'll turn things back over to Simon.
Thank you, Kevin, and thanks to all 3 of you for your terrific presentations. We now have some time for some questions. So if you haven't yet submitted a question to our panel and you wish to, now is the time to do so by clicking on the Q&A box. So let's go ahead and see what questions have come in so far. So here's one. Why does the same sample analyzed for glucose with traditional method RI give a difference of 10% with respect to HPAE-PAD? My apologies.
So I think that question was related to Arcana's presentation. However, from some of the data that I showed you in the last presentation, I think we can probably assume that discrepancies could be due to interferences when the RI detector is utilized. On the other hand, it also could be differences in just inherent measurements made with the 2 approaches. But I think it's more likely that coelution is causing an observed difference in the 2 samples. Okay,
thank you. Another question here is the equipment and procedures, are they publicly available?
So I think most of it is available to my knowledge. Perhaps, Paul, you may need to step in and talk about the availability of the spacer. I don't know if there's a part number for that, for the 62 mil spacer that we routinely use in our lab, but the Reodine valve is certainly available. The current ICS system, which is now the ICS 5000, is available, and it can be routinely utilized in any lab, provided that there's a spacer available.
This is Paul. That's an excellent question. Let me look into what spacer are available, and I'll get back to you later on that.
Thanks, Paul. And I have a couple more here for you, Paul. What are the typical detection limits for your carbohydrate analysis?
Again, I'm sorry. I'll have to get back to you on that question.
I can speak to that, Paul. Okay. You know, obviously, if you're talking about with the larger spacers, I showed some LOQ data in my slide that represented a signal-to-noise ratio of 10. A true detection limit would be based on a signal-to-noise ratio of 3, and in most cases, you're looking at something between 1 and 10 milligrams per liter, but lower limits of detection are not typically the problem in this type of analysis. You're typically trying to analyze samples that have concentrations that are much, much higher than the detection limit that you can achieve with PAD.
Thank you.
Thank you. So there's another question here referring to one of your slides, Kevin, regarding the 62 mil gasket. What is the accuracy at that lower end using this gasket?
It's a great question. I didn't talk a whole lot about it, and I didn't show any accuracy data in the validation study that we were showing today. If you recall from the study we did where we look at response factor as a function of concentration, at the lowest ends, there's pretty significant deviations relative to the average response factor. And so There does appear to be some questions at really low concentrations if you push the detection limits with the large gasket. However, although I didn't show the data, we've done some spiking studies with matrix spikes and have gotten quite reasonable accuracy down to the lower limit of calibration, which was at about 0.2 grams per liter for all the sugars.
Terrific, thank you. I think this is probably another one for Paul. When do you prefer fast versus high resolution separations?
Actually, I'm going to defer that one to Archina, since she has some experience on that area. Archina, can you field that one?
Sure. Yeah, sure, Paul. I can answer the question. So basically, when we are screening a large number of samples to get faster analysis, when we are doing the fermentation samples and we need results like kind of as we go, we prefer fast resolution. And with faster method, we can actually measure sugar consumption at a critical decision point. So then we want to actually quickly gather the data and give the results so that the decisions could be made, faster. However, when we have the optimal separation, we want to monitor sugar consumed at a very accurate level because our yield calculations in the fermentation lab is actually relied on that. In that scenario, we definitely want a robust and very accurate method to be able to define that. And in that scenario, we prefer high resolution. So basically, it would be ideal if you can have best of both. But on a routine basis, I prefer high resolution to be able to provide accurate data on which our yield calculation is based on.
Thank you. So on to the next one. What does one look for in deciding if the fermentation process is or is not proceeding as planned?
Well, so I don't know if any of us are true fermentation experts presenting today, but in general, you're wanting to monitor the decrease of a sugar that's being consumed and an increase in your product level. In a lot of cases, monitoring that product may require an independent column to be utilized. One of the other big reasons that LCRI is used in monitoring fermentation samples is that you can simultaneously look at glucose or xylose and ethanol in a single run. Beyond sort of that introductory level explanation, I think we'd need to talk to a fermentation expert to get the real answer.
Fair enough, thank you. So here's another one. Is it possible that the pulsed aperimetric detector shows different selectivities for different sugars?
Not only different sugars, but different amino acid or neutral sugars. You can dial it in depending on the carbohydrate or moiety of interest. That's one of the unique strengths.
If I might add to that too, Paul, although there are certainly, it's unique to each analyte, it's often desirable that they be similar, and I think if you think back to the calibration sensitivity or the slopes of the calibration curves that I showed in my presentation, the good news is in this case, all of the target analytes have sensitivities that are within a factor of 2, so you don't have large differences in sort of the linear range that could be expected like you would experience with, say, evaporative light scattering or some other approaches.
Thank you. Simon, if I may, there was a question, again, directed to Kevin. When will the procedures for the work that you spoke about be publicly available?
Well, so the methodology for doing this, I'm happy to provide anybody with if they're interested in running the method at this time. It's relatively straightforward if the equipment's available. But we're currently in the process of preparing a manuscript for publication that will show the interlab study and hope to have those in a peer-reviewed journal by early next year.
Okay, thank you. I can add that Thermodynamics has put together an application note on the S8 column with certain carbohydrates that's available on our website if anyone's interested.
Terrific. Well, we're just about to run out of time, but just as a follow-up question to one of our earlier ones, we've got one here saying, does the 62 mil gasket provide a better resolution than the 2 mil? And that's probably going to have to be our last question, I'm afraid.
So I don't know that it has a tremendous impact on resolution. Resolution is largely dictated by performance of the chromatographic column. Obviously, you can get some dilution in the detector cell, but we haven't noticed any substantive changes in resolution of target analytes, going from even a 2 mil to a 62 mil gasket.
That's great, thank you. Well, like I say, unfortunately, we have run out of time there, and so must conclude the Q&A session here. I'd like to thank everyone for attending today's web seminar, and of course, a special thanks to our panel for their presentations. As a reminder, today's webinar has been recorded and will be available for viewing or for downloading in a few days' time. I know a lot of people were asking about that. So in the archive version, there will be a feedback area should you have any further questions or comments.